Our first presenter in the second part of this morning's presentations is Dr. Emily Dangramond, representing Roosevelt University.
Hi, everyone. My name is Emily Dangramond. I am here with you today from Chicago. So I am not enjoying the nice weather you have there in Florida. I'm enjoying sunny, snowy weather. So I'm here to report on a study that collaborators and I have been working on that takes place at the GTM NER. And it's a sort of a follow-up study to an experiment that we had going for many years, or multiple years. So I want to first give a shout out to Candy Feller, who's here with us. She is really the lead behind all of this work, and I have the honor of reporting on it to you all. But she has been really going out, collecting data, coming up with hypotheses, and so she really is owed most of the credit for this work, along with all these other people's names you see listed here. So what I'm going to tell you about today is a little bit of background on an experiment where we added nitrogen and phosphorus to mangroves growing in the salt marsh at GTM. And then I'm going to talk about what happens when that location froze and how that's affected mangrove and salt marsh growth and recovery. So just to orient, you all know where GTM is. This particular experiment was done at the site that we call North Matanzas, because it's north of the Matanzas Inlet. This is also the southern part of the GTM Reserve. And for those of you paying attention to the wet feet talks earlier, this is very close to the NMAT wet feet site. So that site with the most mangroves in that experiment. We set up an experiment starting in 2012 that looked at how mangroves respond to nutrient additions. So this was a site that at the time had mangroves in it, but was mostly salt marsh. And we wanted to know if this site was limited by one particular nutrient. Scrub mangroves in general are often limited by nitrogen or phosphorus, and it depends on the site, but we didn't know what the story was for the site. So there was this experiment set up with small individual mangroves, which were really shrubs less than a meter tall, and they were each assigned to a control treatment or a phosphorus treatment or a nitrogen treatment. So the trees were fertilized annually since 2012. We reported on the results of this experiment in a paper we published in 2020. And what we found is that the site was nitrogen limited. So when plants were given nitrogen, the mangroves just grew a ton more than they did if they were given phosphorus or if they were not fertilized at all. So I'll show you a few graphs to prove that point. You can see the picture here. The nitrogen tree is taller compared to this PVC pipe. In part A here, this is mangrove height. We saw both mangroves and salt marsh plants. So we're really talking about, when I say mangroves, we're talking about Avicennia germinans or the black mangrove. And when we're looking at salt marsh plants in this particular site, we're really looking at Baetis maritima, but there are there's also Sarcocornia there, and there's Spartina as well. It's just not as abundant right in these particular plots. So basically, if you look at these two graphs here, nitrogen is in the middle, and nitrogen plants grew taller and also had larger canopies as measured by their leaf area index than controls or phosphorus plants did. They also, as Nikki mentioned yesterday in her talk in toward the end of the session, is that the growth of mangroves fertilized by nitrogen had negative effects on the salt marsh species. So while the salt marsh species looked like they were responding to nitrogen a little bit in terms of being able to increase their growth, they weren't able to do so as much as the mangroves are. So in this case, the dark bars on the right-hand A plot are the mangroves, the black mangroves. And they've really increased their cover compared to the other, just the bare soil, and then the two different salt marsh species that we were able to detect in our plots, meaning that these nitrogen fertilized trees are really growing quite quickly and suppressing the salt marsh species. And that's resulting in a decrease in diversity as measured by Simpson's D, which is just a metric of diversity in terms of number of species and abundance. So the other kind of interesting thing here is that the nitrogen fertilized plants make more flowers and more propagules, and that could have a big effect going forward in terms of population growth and just the ability of these plants to sort of colonize and grow larger populations in these salt marsh areas. So in January 2018, there was a freeze, and it wasn't just a little freeze, but severe enough that we expected some plant mortality potentially. So we thought, okay, it got down to -4 degrees Celsius on one of those days. Did all the mangroves die? Or put another way, what happened to our experiment? Did we see any differences in terms of the plants that were fertilized with nitrogen versus the control and phosphorus? And we thought that we might see differences. We might actually see more damage in the nitrogen fertilized trees because they've grown bigger, they have grown faster. Theoretically, they would have more tender stems and be just more susceptible or more vulnerable to a freeze. But alternatively, maybe they would suffer less damage because they have these bigger canopies with more branching and increased complexity. And that actually creates a microclimate around the plants where it actually is warmer near the plants. And there is some evidence based on a previously published study that the mangrove shrubs out in the salt marsh are staying a little bit warmer than the rest of the marshes. So let me show you some pictures here. Oops. I lost my cursor. Oh, still lost. Okay, there we go. So what you see on top here is the fertilization experiment in 2016, November 2016, before the freeze. Okay, so you see a lot of green canopy, lots and lots of shrubs, lots of mangroves here. The following photo is in January 2018, right after this freeze. And you can see brown spots in the canopy. These are evidence of damage, okay, freeze damage. You'll see still quite a lot of green though. So our first question was really, are there differences? We know they didn't all die, but are there differences in the freeze damage to the mangrove, to the Avicennia canopy among these different nutrient treatment levels? And then, so we're gonna answer that question, but then going forward in time, a year and a half after the freeze, we actually were able to monitor recovery throughout that period and beyond, even to 2021, and see if there were differences in recovery between these different treatments. So those are the two questions we're looking at here. And I should say this is work that is not yet published, but hopefully will be soon. So the first thing that we're looking at is the proportion of leaves on the left and proportion of stems or woody tissue on the right that are killed by the freeze. And what you'll notice is that in the control treatments and phosphorus treatments, a lot of the leaves were killed by the freeze. So they turned brown and fell off the tree eventually, not immediately, but they turned brown. Almost 80% of the leaves in those treatments were killed by the freeze, whereas the nitrogen trees had very low percentage of leaves killed by the freeze. Similarly for stem damage, overall there was less stem damage than leaf damage, but there was stem damage in controls and phosphorus, and none of the nitrogen trees had any stem damage, which is pretty incredible. If we look at then that same thing I just showed you in the first graph with the proportion of leaves, and think about how it's related to other things like tree height and canopy area, there is a relationship there where we know that the nitrogen trees were larger and they also suffered less stem damage or leaf damage and stem damage. But the slope of these is about the same. So the relationship doesn't really differ too much, even though the nitrogen trees were so much larger. The control and phosphorus trees had stem damage, had leaf damage, and it's not necessarily a direct relationship with tree height. And this looks similar for canopy data as well. So what we did to try to disentangle this a bit more with all these things we've measured is we did what's called a principal component analysis. So we have all these potentially correlated variables, and we do an ordination to figure out what's kind of related here. And what's interesting about this graph, what should jump out to you, is that the nitrogen plants, which are shown in blue triangles, those are separated out from the control and phosphorus plants. So all the things that went into these sort of explanatory variables for this PCA in terms of total freeze damage were things like plant height, canopy growth, leaf area index, percentage of carbon and phosphorus and nitrogen, glycine betaine, which is in osmolite that's thought that protects plants against salt and may have some protection against freeze as well. So the major takeaway from this is that that horizontal axis, PC1, had mostly canopy level traits like plant height and canopy growth. And that vertical axis where there's more overlap between the different plants had more leaf level traits like nutrient content, specifically phosphorus and C/P ratio and glycine betaine. So we know that the nitrogen plants are doing something different, and that somehow they've managed to survive this freeze a little bit better, come out of it with less damage. What was interesting there is then we started to look at recovery, okay? And I'm only gonna show you one graph here because they're a little bit complicated. There's just multiple years. This is actually the proportion of shoots flowering, but we also have growth data and shoot elongation data. So remember, 2016 was before the freeze, okay? So we knew that nitrogen plants in blue were flowering more, they were growing more, had longer shoot elongation, larger canopies. Then if we look at what happens after 2016, so in 2018, that was the year of the freeze, there was a decrease for everyone in terms of flowering. And then after that, so in 2019, then 2021, we actually saw increases in those controls and phosphorus trees that made them start to look more like the nitrogen trees, or at least were similar to what they looked like in 2016. And the graph I'm not showing you, but you're just gonna have to believe me, is that the growth data looks in 2018, right after that year after the freeze, the control trees and phosphorus trees look very similar to the nitrogen trees. So they started elongating and growing quite quickly compared to what they had done before. And that's kind of strange because they had freeze damage, right? So I will show you a picture instead of a graph because I think the pictures are almost as good, maybe not quite as good, but yeah. So we can look at this is 2016 on top, the 3 different treatments. 2018, after the freeze, you see a lot of brown canopy and the control and phosphorus and a lot of green in the nitrogen. And then by July 2019, we see total recovery. So you can see there's just nice bushy plants with lots of leaves, and there's been a lot of growth in that period as well. And you may be thinking, okay, so we know that these plants had more damage, right? How on earth did they recover so quickly? So this leads us to this interesting hypothesis that we are gathering data for, that we believe we know that there were freeze-killed leaves on the control and phosphorus trees in high abundance. After the freeze, those leaves that had been killed fell to the ground and decomposed as leaf litter does in mangroves and marshes. What's different about this time around is typically mangroves will pull nutrients from their leaves and then drop them as yellow senescent leaves that are nutrient poor by the time they fall to the ground and decompose. So the leaves are not necessarily, they are contributing to the organic matter in the soil, but they don't have a whole lot of nutrients left for the plant. What's different about this time around is that in 2018, during that freeze, the leaves were mostly green on the plant. Then they were killed by the freeze, they fell to the ground, and it's now nutrient-rich leaves falling to the ground as leaf litter, potentially providing a source of nutrients for those plants to then absorb through their roots. So this is something that we're still investigating. We have some preliminary data from experiments that supports this hypothesis that the trees sort of went through a self-fertilization event, if you will, and that allowed them to recover very quickly, is what we believe happened. So I will, let's see. think, sorry, that's my last slide there. So I will leave it there and see if anyone wants to ask questions.
Wonderful. Thank you, Dr. Dangramond, for that wonderful presentation. Does anyone have any questions? I don't see any in the chat box quite yet, but we have plenty of time for questions.
Candy has previously shared a similar slide full of graphs, so I guess that would have occupied us a little longer, but I don't want to get too crazy.
Okay, we have some questions here. Did the small shrubs regrow from the base, or were they killed outright from the freeze events?
Okay, so this is a good question. Thank you. So I actually we really didn't see the plants actually die. Okay, we saw mortality of stems, but the actual plant themselves didn't, like, there wasn't, they survived the freeze. Their stems, maybe some of their stems died, their leaves died. So they did start to regrow from the base. And this is another piece of data we have, is looking at this resprouting that's happening. And we've documented in, and actually the same site just nearby, that if you kind of go down in the mud and feel around for past stems on those shrubs, you'll find that there were previously stems that probably were killed off by the freeze and then they resprouted. So this is another question that we're looking into, is, are these plants really just resprouting, potentially contributing to their shrubby form? Are they reproducing more after the freeze? Are some plants reproducing more by propagules, and some plants are kind of reproducing by, or resprouting from epicormic shoots? And it looks like, well, I guess I don't know if we have a conclusive answer to that question yet. So I'll just leave that for the next question.
Thank you. How was the nitrogen applied, and in what form was it?
Yeah, so nitrogen was applied as urea, and it was applied via dialysis tubing that is basically dug into the ground as a little soil core plug is removed, and the dialysis tubing and urea is put down into the ground at the base of the shrub, and that's done annually. So this is really targeted fertilization treatment. It's not broadcast throughout the whole site. It's targeted. We're pretty confident that the fertilization treatments really only affect the target plant and maybe about a meter or radius around the plant. So we know that our treatments are getting to the plant that we're studying and not getting everywhere else.
Great, thank you. How do you think self-shading from radiation plays into this?
Okay, that's a good question. How does self-shading from radiation play into this? So plants and mangroves can certainly have too much sunlight, and they can shut down their photosynthetic machinery if that happens, like in the beginning of the day or middle of the day when the sun is really bright. But of course, light is a limiting resource as well, or they need light to grow. So the larger plants would potentially have more self-shading. I don't know. That's a good question. I'd be interested to hear if anyone else has thoughts on that, but we could imagine that there's more shading going on in larger plants, although these plants are growing very much out and not as much up, right? So they are probably trying to limit their self-shading, although they are likely shading some of the recruits under them, the seedlings under them. So I don't know. We'll have to think about that one. But we know that microclimate can be altered. But I haven't thought so much about shade.
Awesome. Well, we have time for maybe one more quick question. Is it possible that some of the nitrogen added mangroves, is it possible that that recovery could maybe be explained by greater storage of energy in their root systems?
Yeah, that's it. So, right, that's a good question. So so someone, let's see, was it Pam earlier was talking about how when plants are fertilized, they maybe don't grow their roots as deep because they don't have to search those nutrients out, right? So would there potentially be an interaction here where roots in the non-nitrogen plants are deeper maybe and have more energy stored and then can recover? That's certainly a possibility. We haven't really explored the below ground story that much in this fertilization experiment. It's hard to do. You can't do it without some destructive harvesting. But that is an interesting idea. And we're actually, and to sort of answer another question that came up, a master student who's working with me now is doing a lab experiment where she's growing seedlings that have been fertilized with these different types of leaf litter. So frozen leaf litter or like green leaf litter, senescent leaf litter, to look at how above ground growth and also below ground growth respond. So we might have an answer, a somewhat answer to that question soon.
Wonderful. Thank you, Dr. Dangramond.